Let me tell you an incredible story of scientific persistence. It's a story that begins not in some sterile, high-tech lab, but out in the wild, deep inside the natural world. I know, it sounds like something out of a fantasy novel, right? But back in the middle of the 20th century, this was a very real and a very urgent question that kicked off a scientific quest that would last for decades. So, to really get why this was such a huge deal, we've got to rewind the clock a bit to a time when the entire landscape of medicine was changing, and fast. Take a look at this timeline. It shows how a huge medical victory actually created a brand new challenge. See, thanks to the miracle of antibiotics, people weren't really dying from infections like pneumonia anymore. But as people started living longer, they began running into new enemies: heart disease and, you guessed it, cancer. So in 1955, the National Cancer Institute decided to do something pretty radical. They launched this massive public screening program. The mission? To hunt for new anti-cancer agents anywhere they could possibly be found. And by 1960, their focus zeroed in on the planet's biggest, most unexplored chemical library, the plant kingdom. To spearhead this ambitious hunt, the NCI brought in the brand new Research Triangle Institute, or RTI. And this is where we meet the heroes of our story. The lab was led by a chemist named Monroe Wall, who in 1962 was joined by Mansukh Wani. This duo, well, they formed a scientific partnership that would completely change the face of chemotherapy. And you know, their search for a cure wouldn't lead them to some exotic, mythical plant. No, it led them to two pretty ordinary-looking trees, each holding its own chemical secret. From the outside, they were just trees, just bark and wood. But inside, each one contained this incredibly complex molecular library, fine-tuned by millions of years of evolution. So the challenge for Wall and Wani was to find that one molecule, that one secret, that could actually fight cancer. All right, so let's meet our contenders. On the left, you've got Camptotheca acuminata, a tree from China. An old sample of this one showed powerful anti-cancer activity right out of the gate. It was an obvious lead. Then on the right, we have the Pacific yew from the U.S. Pacific Northwest. Its bark showed some cell-killing activity, sure, but it wasn't nearly as dramatic. So you've got one that looks like an instant superstar and another one that's, well, a maybe. With the samples in hand, the hunt moves from the forest into the lab. And this, folks, is where the real grind begins, trying to find that one active molecule hidden among thousands of others. The process they used is called bioactivity-directed fractionation. I know, it's a mouthful. But think of it just like panning for gold. You start with a big bucket of crude extract, all the chemicals from the plant mixed together. Then you start separating it into smaller and smaller groups. You test each little group to see if it fights cancer. If it doesn't, you toss it. If it does, you keep it and purify it even more. You just repeat this over and over and over again, until, if you're lucky, you're left with a few flecks of pure chemical gold. And you won't believe it, with the Chinese tree, it was like they struck gold on the first try. They isolated the active compound, they named it camptothecin, and by 1966, they already knew its chemical structure. The NCI was ecstatic. In their own words, this project was very hot. It really looked like they had found their miracle drug. But the Pacific yew? Oh, that was a whole different story. Isolating its active compound was brutally difficult. In fact, their work was so slow and everyone was so excited about camptothecin that Monroe Wall was this close to just abandoning the whole yew tree project. But right when it looked like they had a clear winner and a total dud, the whole story gets flipped on its head. Both of their amazing discoveries were suddenly and shockingly on the verge of complete failure. That very hot compound, camptothecin, it completely bombed in early clinical trials. It turned out to be way too toxic for patients, and it was nearly impossible to dissolve in water, which made it an absolute nightmare to turn into a usable drug. Just like that, the most promising anti-cancer agent in years was put on the shelf, and the research just sat there for almost 15 years. In the meantime, after years and years of that painstaking work, Wall and Wani finally isolated the compound from the Pacific yew. They named it Taxol, and it looked incredible. But then they slammed into a massive wall, and that wall can be summed up in one staggering number: 360,000. That's how many of these rare, slow-growing trees would have to be killed just to treat ovarian cancer patients for a single year. It was an environmental and logistical impossibility. So here they are. Two miracle compounds. One's too toxic, The other's too rare. It seems like a dead end, right? Well, the breakthrough didn't come from finding more trees or trying to tweak the formulas. It came from asking a much smarter question. How did these things actually work? This brought them to a concept that's absolutely critical: the mode of action. Basically, it's not enough to know that a drug works, you have to know how it works. What's the specific weapon it's using to fight the disease? Figuring that out, well, that was the key that unlocked everything.
And what they found was brilliant. The two drugs worked in completely different ways. Scientists discovered that camptothecin works by trapping a key enzyme that cancer cells need to replicate their DNA. Imagine the cell's DNA is like a zipper that needs to unzip to make a copy. Well, camptothecin just jams that zipper shut. No replication, no new cancer cells. Taxol, though, was wild. It went after the cell's internal skeleton, its scaffolding. But instead of breaking it, Taxol made the cell build so much extra scaffolding that it literally got clogged up and couldn't divide. These were two totally new ways to kill cancer. And knowing how they worked, that was the game changer. For camptothecin, chemists could now design slightly modified versions, called analogs, that were way less toxic but still kept that zipper jamming ability. And for Taxol, they found a way to make it in the lab using a molecule from the needles of the common English yew, a totally renewable resource. The supply crisis was over. The weapons were finally understood, and now they could be perfected and mass-produced. And so after all those years, all those dead ends and amazing breakthroughs, two humble trees finally made good on their promise, creating a legacy that is still saving lives to this day. You just can't overstate the impact. What started as a hunt in a forest has led to essential treatments for ovarian, breast, lung, and colon cancers. As you can see here, we now have two totally different drugs from two different trees using two totally unique mechanisms. And both have become powerful, essential weapons in the war on cancer. And finally, in 2003, this whole incredible journey, from that first screening program all the way to the final drugs, got the recognition it deserved. It was officially named a National Historic Chemical Landmark. And really, the words on that plaque they put up say it all. This wasn't just about finding chemicals in trees. It was about discovering entirely new ways to fight a disease. It's an amazing reminder that sometimes the most important answers are hiding in the most unexpected places, if we just have the persistence to keep on looking.